Hi everybody, my name is Hennie Groenewald. I'm the manager of Biosafety South Africa. We're doing a new thing today. I presented 1 million lectures in my life. This is the first time that I'm standing alone in my office staring at the computer screen. So it's a little bit intimidating, but hopefully we can use these new technologies and new ways of communicating with people to reach a broader audience. And this is one of the objectives of Biosafety South Africa, the organization for which I work. It's one of the objectives it has. So I've made a few slides, and I just see that I can't actually show the slides when I'm live, so I will have to explain everything to you. Hopefully that works, and this is the way that we learn. So starting off by introducing Biosafety South Africa, just quickly, it's a national service platform. It's funded by the South African government. It's under the auspices of the National Department of Science and Technology, and we are hosted within the Technology Innovation Agency. The platform's mandate is to support sustainable biotech innovation. I think the emphasis lies on the word sustainable, which means everything we discuss related to biotechnology is discussed within this context. Just briefly, sustainability has 4 legs on which it stands. It's in the first place the food and feed safety issues. The second is the environmental issues, and both the food and feed safety and the environmental safety contributes towards biotech products, biosafety. When I refer to biotech products, maybe you can specifically think about genetically modified organisms, which is one of the biotech products that we get. We see that most often in the media. It's products that are actually on our markets, the type of things that people want to know more about. So coming back to sustainability, we just said the biosafety is the health and the environmental issues. And then there's also the viability issues, which relate to the socio-political and then the techno-economic issues. Socio-political means basically that you can't apply or use regulated technologies in an environment where you don't have a regulatory framework. So in South Africa, for GMOs, for example, we have a GMO Act, which specifically governs all activities with GMOs. But then there's also alternative or additional regulations under, for example, the Department of Environmental Affairs and the Department of Health and the Department of Trade and Industry, things related to labeling and environmental sustainability and all of that. And then the fourth leg is the techno-economic issues. It basically just means, obviously, if you want to develop a product, you need the technology to do that in the first place, and then it must make economic sense, because in the end of the day, biotechnology is applied science, and it's only applied when there's a product, and there's only a product if there's a market for it. So then what we're actually going to talk about today is what we refer broadly to, especially in the plant breeding environment and agricultural related applications. We talk about new breeding technologies. So one of the points that I want to try and clarify today is, in the first place, to say that a term like new breeding technologies is actually very wide. To really start talking about things that we will touch on later, things like ethics, issues like safety, we can't just talk about new breeding technologies because it's just too wide. We need to narrow it down. So one of the components of that is something specifically referred to as genome editing, and this is then also what I will predominantly focus on today. So I've got a very nice slide here to my left, which unfortunately now I can't share with you, but it just shows some of the headings, and not even headings. Lots of the very prestigious scientific journals had front pages that says things like the DNA revolution, CRISPER-CAS, which you may have heard of, is the world over. It's very powerful technology in terms of how you can edit the genetic material of living organisms. So then, quickly, I just said new breeding technologies is a very broad term. So the Academy of Sciences of South Africa in March last year published a consensus report on looking at these new breeding technologies and some of the technical and the risk associated aspects of that, and then also trying to discuss and propose how we should then look at the regulation of these things. And I'll come back a little bit later to the regulation part again. So just quickly, new breeding technologies, we've divided it in broadly in the genome editing or targeted mutagenesis techniques, which is the major group, and it's predominantly the one that we will discuss today. But then there's also the so-called null segregants, which just means you use a regulated technology, or in the case of GMOs, you use a GMO in the breeding process. But then later on that genetic modified piece of DNA, the transgenic material is actually taken out of the equation. So it's not in the final product. That's why it's called a negative or a null segregant. Examples of how this is applied is, for example, to change the rate with which a plant becomes sexually mature, like trees and plants that grow over multiple years. Usually there's a long time before they can become reproductive. And of course, in a breeding program, you're dependent on this, and you want them to become reproductive as soon as possible, because then you can quicken the breeding process. So you can do that by having a transgenic plant, for example, which will mature quicker, do your breeding, and then you segregate your transgene from that. So you do the breeding program, you select the plants that you want, but the ones containing the GM traits then are separated. And then the last group, so we already done the genome editing or targeted mutagenesis, which I will come back to because it's the most important one. I just spoke about the null segregants or negative segregants. And now then there's a whole other group that's actually just variation of genetic transformation. So traditionally, everybody knows about genetic modifications that leads to the production of genetically modified organisms. And there's obviously a lot of detail we can discuss about exactly what is called a GMO and what is not. The bottom line is that these things are defined by laws within the different countries. In South Africa's GMO Act, a GMO is defined as an organism which contains genetic modified DNA that could not have been generated in a natural way. So basically what that means is when you use DNA from a different organism that cannot cross with the plant or the GMO that you made. So for example, if you have maize, and sorry, many of the maize plants we have a bacillus, which is a bacterium gene in it to make it insect resistant. So naturally, you can't cross a plant and a bacterium. And that is then actually the threshold or the trigger for regulation, is because you have this novel combination of DNA. You can also have variants. People refer to things like cisgenesis, for example, where you use DNA from the same species. So these are species that could cross naturally, but instead of doing the crossing, which can lead to lots of other complications that we won't have time to go into, but you can then take from a different species. You can take a piece of trans DNA and then using molecular biology and gene cloning technique, you can transfer that DNA. You can do it even a further refinement of that is to do something which is called intragenesis, where you actually take DNA from the plant itself and you just manipulate it somehow and move it around. Doing this can lead to a lot of changes in gene activity, which in the end translates to changes in the phenotype or the characteristics of the organism. And then finally, there's also one that's called transgrafting. In the Western Cape, for example, all the wine grapes are planted on rootstock that are resistant against specific soil-borne diseases. So we need that. If you just plant a pinotage or a merlot, the plant will be attacked by the pathogen. But if you inoculate it on a resistant rootstock, they are safe and they grow well. So the idea here is to say, instead of doing that transfer, you can now take a transgenic root that or rootstock that will give you a lot of different characteristics that you are looking for, and plant the top part of the plant. But that now will not be transgenic. So this is this discussion, for example, if it bears fruit or not. It's completely different vegetative material. So obviously, if you look at it from a legal perspective, there's a lot of debate, but biologically speaking, that's a completely different plant, so it's not transgenic. But in terms of regulation, we have to sort out these things because those are legal systems. So quickly coming back to the genome editing techniques or targeted mutagenesis, these are all basically, they call targeted mutagenesis because you actually can generate a tool which in the first place would recognize a very specific target sequence within your genome. And then to that tool, you also add a new piece of DNA, for example, that you want to insert, or you don't even have to do that. The bottom line is you need a target sequence that will align with the sequence within the cell that you want to change. And then you need what we refer to as a nuclease activity, which is basically an enzyme that will cut the DNA. And then you use the internal mechanisms of the cell itself to repair that DNA. Every organism, including you and me, have in each cell of our bodies, there's enzymes that repair DNA. So if you're exposed to high energy radiation like UV light, for example, it can damage the DNA in your skin cells. And this is why cells have mechanisms to repair that DNA. So doing genome editing in this way is a combination of using the mechanisms and the tools that are available in the cell and then combining them in a way that we can use them very specifically as tools to change DNA sequences with. So again, and I want to remind you that I will post all of these slides on Facebook afterwards, that you can refer to these. We will place them as photographs, then you can refer to them. But the DNA, then the types of changes that we can make with these genome editing things is we can change a single nucleotide, for example. There's diseases in humans, for example, things like osteogenesis imperfecta, which is a genetic disease that's caused by a single nucleotide change. So what we can theoretically then do is target that piece of DNA and change that single nucleotide to repair it. You can also modify it. If you talk about enzymes, for example, you can increase the affinity for the substrate. You can change the way that that enzyme interacts with inhibitors and things like that. So you can change metabolic flux through making very small changes in the DNA sequence, which is then translated to the protein or the enzyme that I just referred to. You can also delete pieces of DNA. You can insert whole genes or not. So there's a lot of different ways of doing that. So please refer to the slide that I have. We also have an infographic with the title Genome Editing that will give you all that detail. So why do we want to talk about genome editing? It's because it can be applied. So I've got a short list here just to give you an idea of the type of research that our people are doing. And I've got 3 marked on my slide here that shows which products have actually already been gone through to the market. The first one is a non-browning mushroom. You know, these white button mushrooms that we often get in South Africa as well. When you cut them and leave them a while, they brown. That brown is a natural reaction of the cells that you broke when you cut it. But that's an enzymatic reaction. So just like I explained earlier, one of the things that you can do is to inhibit the enzyme that causes that browning. So in other words, you cut the mushrooms and you can leave them on the shelf for a while. If a producer, for example, wants to cut or sell cut mushrooms already, you won't have a lot of brown mushy mushrooms waiting for you on the shelves in the shop. And then there's something that's already been approved. It's a little bit of a technical issue. It's actually not been approved because it was decided in the US system that it does not fall under the regulation, which I will talk about earlier, but it's on the US market already. And then there's a herbicide-tolerant canola. Some of you that may know of the GMO crops that are in South Africa, you would know that herbicide tolerance is one of the important traits that are in GM crops in South Africa because it allows you, for example, things like minimum tillage and protection or environmental sustainable agriculture. So there's now a herbicide tolerant one where, remember, herbicide is a molecule or a chemical that actually binds to a particular enzyme, for example, and it stops the activity of that enzyme. And if that's a critical enzyme in a important metabolic pathway within the plant. For example, some of them will inhibit protein synthesis. The plant, with the chemical there and the enzyme activity dead, it can't produce proteins, and therefore the plant will die. That's then a herbicide. But again, that interaction between that chemical and the enzyme is very specific. So if we can change the protein, that the chemical doesn't bind to it, or we can inactivate that chemical, and somehow you have herbicide tolerance. There's also many other examples. I think things like cabbage that grows slowly, vitamin-enriched oranges, drought-tolerant maize and wheat and rice, all projects that are at the research phase. Then there's also animals. There's a big project in the US looking at hornless cattle. So the argument here is when you have cattle on a farm in very close proximity, they can hurt each other with the horns. So you may even know that naturally you get cows or cattle without horns naturally. So what biology has done first is to characterize that trait and discover that it's a particular gene activity. So now if we understand how that works, we can transfer that trait to other cattle breeds that do have horns, and we can have hornless cattle. There's also technology that's looking at determining the sex of animals, things like female chickens only. Then you don't have to get rid of all the male chickens, for example, if you're interested in producing eggs. And then another very interesting technology that's already been applied actually is gene therapy in humans. But I will leave that a little bit because I have a very good discussion right at the end of the ethics of applying these techniques to humans, and then I will refer to this again. But the point is that this technology has already been applied on humans in therapeutic treatments. And the specific example I have here is the one of a little girl that was treated in the UK a few months ago. So then I think quickly the other point I want to make, you'll remember that I tried to, in the first place, make a big point about the fact that new breeding technology is a very wide group of different technologies. The point I want to make now is those are the techniques that you use. So what I often come across is that we talk about a technique like genome editing, and then somebody will ask questions about another term that they heard, like things like gene drives. So before I explain gene drives a little bit, the point I want to make now is to distinguish between the technology that you use, the impact that that technology will have on the genetic sequence of the organism that you're busy changing or introducing variation in its genetic material, and then you apply it in a certain way. My other hand is low here. So the point is we must distinguish those three different facets of applying genetic variation technologies. Because I think one of the important points is to say when we do a risk analysis, the risk analysis is the analysis that you do when you produce a GMO, for example, to say, what's the likelihood of having an impact on the environment or on human health? Then you go through the whole process, which we can discuss in a different time. But when you do a risk analysis, you don't only look at one of these aspects. You can't just go and say, it's CRISPER-CAS that I'm using, and therefore it has these implications. You can use CRISPER-CAS, the technology, for example, to make a single nucleotide change in a genome, like I explained earlier, or you can use it to insert multiple copies of genes from different organisms. In other words, you can make changes in an organism that is exactly and identical to those changes that are naturally occurring within organisms. This is why we see variation among animals. It's a natural phenomenon that genetic sequences and traits change over time. That's actually what evolution is based on. But then on the other side, you can use the same technology to insert genes that cannot integrate into that particular organism through natural means. In other words, things that we call genetically modified organisms. And then finally, you have the application. So those applications, we have harnessed genetic variations for hundreds of years through selection and breeding. But on the other hand, we have now technologies, something like gene drives, for example. Gene drives is a way of changing not only the genetic trait of the organism, but also changing the way in which that DNA is inherited by subsequent populations. So it's a technology that will allow us for the first time to actually go and impact on wild populations. So obviously you can hear from that the risks associated with these things are completely different from others. So the way then in which you, for example, if that was a single gene, you can either look at this as a GM crop like the maize that we have at the moment on the one side, or you can say this is an organism with a gene drive technology. The way in which you apply the technology, for example, have a huge impact on its potential risk. And this is why we should consider these things always on a case-by-case matter. So then, the point that we made in the ASAF report that I showed earlier, I already tried to explain this, but when we consider regulation, I think the first thing that we should think about is why do we regulate things? You can think of GMOs that we talk about now, but you can think of something as simple as driving a motor vehicle on the South African roads. We regulate it because there's a risk associated with it. If everybody were allowed to drive as fast as they want without having the necessary skills to drive, in other words, having a license and things like that, where people make sure that you can drive, then we will end up with a system that has a lot of risk in it. The other example is something like, the roadworthiness of your vehicle. If somebody drives at very high speed on the N2 and they can't handle the motor vehicle because one of the wheels is coming off or it doesn't have brakes, it can have huge impact. Long story short is we regulate it. So GMOs, I just made a point earlier, why do we regulate GMOs specifically? Is because for the first time we are capable of transferring genes and proteins. We call it a context in which they were not before. So we've always eaten maize. And why do we know maize is safe? The only reason there's no research been done on maize originally, it's called history of safe use. So we eat maize and we know it's safe because we've eaten it for hundreds and hundreds of years. But now for the first time I have the power to transfer a protein from a bacterium into the maize. And now suddenly I can ask, but what if this protein gives an allergenic reaction? And this is why we regulate it, to give us the opportunity to answer those questions. So that's what regulation is about. Basically, regulation is to manage risks. So coming back to my whole explanation of new breeding technologies, the various technologies that can be used from anything from making small changes, like we will see naturally occurring in organisms, all the way to huge changes like introducing novel genes. It means that we have to put in a threshold somewhere. It does not make sense to regulate a risk that is equal to the risk that occurs naturally. If I now cross two different strains of maize, the population will have a huge variability. Just like when a mom and dad has different children, they look completely different because they inherit different compositions of the genetic material from the parents in the first place. And then there's actually mutations that happen within the gametes or the sex cells in the parents. So all of those changes are transferred to your offspring. And this is why you have millions of potential variants just between two parents. So the point I want to make is, again, connecting risk and regulation, is to say, as soon as that risk is beyond the point of the risk that we expose to naturally, then we should regulate it like we do regulate GMOs. But if that risk is equal to what we experience every day, like I just tried to explain, if you make a cross of a tomato in your backyard, every seed is an individual, a unique individual. One of them may have an upregulated protein which tends to be allergenic. So literally, when you plant those different seeds, you can be allergic to the new fruits from the one plant, but not to the others because of the naturally induced genetic variation. So the point we make is to say, in terms of these new breeding technologies, we should regulate those things that have higher risk that occurs naturally, and we should not regulate the others. So Now, the last point I quickly want to touch on is the ethics, because there's a lot of discussion depending on what you're exposed to and what your background is, but it's not only in the general news media. There's also lots of academic discussions about the ethics of genome editing, but most of those are regarding very specific applications. So one of the applications I already spoke about, it's something called gene drives, where I said a gene drive can be used not only to transfer a particular trait, but also to impact the way that genetic material is inherited. So usually you all know that kind of Mendel's old story, that if you have a white flower and a purple flower and you cross them, you can get pink flowers. It's more or less middle of the way. It's a very simplistic view, but this is more or less how it happens. What happens with that is that in that new pink generation, you have one white gene and one purple gene. But if those now go on to the next generation, the individual, although it has pink flowers, it only has the gene for the white gene. So the point I'm trying to make is that the characteristic that genetic material segregates. So in the end of the day, traits disappear along the way, or they become amplified depending on the environment to which these organisms are exposed to. But if you have a gene drive, you force the inheritance. In other words, every individual will have, if you put the gene drive next to the purple gene, every one of the offspring will have the purple one. So why is this long story important? It's because what we can do now is, for example, change the sex of an animal. Let's think of a mosquito. So only the females transfer diseases like malaria. So what you can do is you change the population, or you introduce to the population mosquitoes, or the gene drive that will change all the mosquitoes to males. But you shouldn't stop thinking there just to think, oh, males won't transfer malaria. What will happen if there's only males in the environment? There will be no females to pair with, so the whole species will be eradicated. So obviously, again, why is this a controversial application, a potential controversial one? Because you can actually make a very specific species go extinct. You can design it to go extinct. If you just look on the surface of that, obviously it does not make sense, and we should not do things like that. But if you consider that there are more than 3,000 species of mosquitoes in the world, and only 3 of them can transfer malaria, and you argue millions of people are dying of malaria, and you take out those 3, you understand in the end of the day, it's an ethical question. Do we have the power, or should we eradicate a complete species? And we can have, like I just tried to show, there's pros and cons. On the one side, for the malaria mosquito, it's good for the people, but on the other side is we as humans make a decision about the extinction of a specific species. And these are open-ended questions. There's no way that we can answer ethical questions with just data. The other point I quickly want to end off with then is genome editing in humans. Again, here I've got a nice slide, which I can't share with you, but which I will share on Facebook afterwards. But again, that it's not a simple thing. When you say genome editing, usually if you just read what's going on in the papers, you will think genome editing, humans, designer babies. But designer babies, or what people call designer babies, is only one of the aspects. What we must think of in the first place is when you do genome editing within humans, you can either do it only in their somatic cells. Somatic cells is the cells which makes you as an individual. So it's everything in your different organs and tissues. So if you have, like I explained earlier, the baby girl in the UK, they will take stem cells from your bone marrow, for example, take it out and then change it or edit it, the genetic material of that, to correct the mutation that's in there, and then they put it back in the somatic cells, or in the stem cells, will then give rise to things like blood cells, for example. And these will be healthy ones, so you will treat the disease. So there's no difference actually from these than from any other therapeutic use of any medicine or drug. But then you can also, the point I must just quickly, sorry, I'm interrupt myself. I just want to say the other thing that's very important to know is if you then replace these cells and it becomes blood cells, that is only part of that individual. You don't impact the children of that individual. Then you can also, let's first look at somatic cells and then germline. So in the somatic ones, I just spoke about the therapeutic use. In other words, we cure diseases. It's medicine. Then there's another level in which you can use somatic cells, is where you change somatic cells, but it's not because it's a disease. So you can equate that to something like plastic surgery. So it's enhancement in somatic cells, but it's only the individual in which you edit the DNA that it's enhanced. It does not go to the children. And then the second type of changes that you can make is in the germline. So these are the egg cells and the sperm cells, for example. The point is you change DNA there, these are transferred to the next generation. So it's not only, you don't make a decision to change only something in your own body. You're actually making that decision for prosperity and for your children, your children's children. And again, it's clear why this is an ethical issue. You have to consider that you're going to change, and this change that you introduced is going to be influence in perpetuity. So the question is, first place, just quickly referring back to my other two, you can see it's very difficult explaining this without the help of my slide that I designed. But in the first place, we can use that as well as a therapeutic way, where we can now, for example, make sure that our children won't be sick. But then the other one is if we use germline and enhance them, this is where we get to the tricky part, where they say designer babies. So it's not about eradicating a disease that you will transfer to your children, but it's to give them blue eyes or, long, beautiful hair. So that's why it's an ethical issue. But the point why I discuss these in the first place is, of course, interesting because for the first time, humanity has in its hand the power to determine its own evolution. So it's obviously, a shedding moment, a water-shedding moment in our history. And that's all I have to say today. This video will be available on our Facebook page, as far as I understand. Like I said, we will share the slides, so hopefully people can catch up. And we've learned a few things this time, so hopefully next time we will incorporate those and give you another interesting talk. If you have any questions, please drop them in our inbox and we will continue discussion on Facebook. Thank you.